This video talks about different types of amyloidosis. Now, amyloids are insoluble proteins, and something went wrong in their secondary structure. As a result, they start depositing all over the body in different organs. Okay. Now, there are 7 different types of amyloidosis that is mentioned here. These are the most commonly tested. Okay. So let's first start with Benz-Jones type. Benz-Jones amyloidosis deposits something called AL, okay, and these AL protein are derived from immunoglobulin light chain. And another way you can kind of remember is that AL has the L in it, and L can stand for light chain. Now let's talk more about the Benz-Jones type of amyloidosis. Now, these famously Benz-Jones proteins are monoclonal globulin. Okay, monoclonal. Mono means one, clonal means clone. So monoclonal is that they're just making that one type of immunoglobulin more, a lot more than the other types. That's why it's abnormal, right? It's making that one clone. Another way of calling them is immunoglobulin light chains. All right, so again, the L stands for the light. It doesn't stand for light chain. It's a memory thing. It's easier to remember the Benz-Jones proteins. Okay, so let's quickly take a quick overview about the structure of an immunoglobulin. So we have the heavy chain, the light chain, and then this is the antigen binding site, right? This region is the FAB. This whole thing is FC, and FC can be divided into 2 regions. There is the complement binding site and the macrophage binding site. So complement comes first, so C for complement binding site, and the second region is the macrophage binding site. So in this question, how does it relate to our Benz-Jones proteins? Because Benz-Jones protein is only making these light chains, and we're calling them Benz-Jones, right? We see them often in multiple myeloma, right? We see lots of multiple myeloma. Benz-Jones proteins are monoclonal spike. We see that monoclonal spike due to the immunoglobulin light chain. We also see it in Waldenström, right? How do we differentiate between multiple myeloma and Waldenström when both of them are making the same kind of immunoglobulin light chain? Remember that in multiple myeloma, we're going to have bony lesions, right? We're not going to have bone lesions in Waldenström, so no bone lesions. Now, since we're talking about this, let's talk about multiple myeloma a little bit more. Okay, first of all, there is heavy chains and light chains, and there is 2 types of light chains: the kappa light chains and the lambda light chains. Okay, and these are the only 2 types, and don't mix the lambda with other signs. That's what the lambda looks like. It looks like the Y being an upside down Y. Okay, so these are the light chains. But when we look at an electrophoresis, we see that this is the albumin spike. This is alpha 1, alpha 2, beta 1, and gamma. So usually gamma is supposed to be a small spike, but in multiple myeloma or in Waldenström, we're going to see a big spike, a big M spike, a big monoclonal spike. And the reason we see this big monoclonal spike is because in multiple myeloma, we're making lots of IgG and a little bit of IgA, okay? But more IgG, immunoglobulin G. So that gamma is kind of showing up in our gamma spike, our monoclonal spike. So that's why the spike goes up in multiple myeloma. So that is all that we need to know about Benz-Jones proteins. Now let's talk about histology a little bit more. With multiple myeloma, we're going to see this typical periorbital hemorrhage. And we're also going to see kind of thick tongue due to the amyloidosis. So these are some of the common symptoms that you can see with multiple myeloma, and deposition of our AL protein, our Benz-Jones proteins. Okay, so it's going to be depositing everywhere. So these are some of the things we're going to see physically when we do a physical exam. Now the next one that we will be talking about is secondary amyloidosis. Secondary amyloidosis has deposition of AA, 2A proteins, and they are serum amyloid associated. Okay, and usually we see secondary due to inflammatory process. So lots of inflammation cause secondary amyloidosis. Now serum amyloid associated, or SAA, is what serum amyloid associated stands for. The A is coming from acute phase proteins, and this abnormal AA is been derived from this serum amyloid associated protein, right? And I also already mentioned that this is due to inflammation and due to acute phase proteins. Now, you can remember acute phase by the A's that you see here. There's lots of A's, so acute phase. So let's talk about acute phase a little bit in detail. So really, there are two types of acute phase reaction. There's the positive acute phase protein and then the negative. The positive one deals with when there is an inflammation. If proteins are released due to that inflammation, that would be positive acute phase protein. And negative would be due to inflammation. If there is decreased release of a certain protein, that effect would be a negative acute phase protein. So what is interesting about liver is that liver responds by releasing acute phase proteins, okay? But as a result, it suppresses other proteins which are also important for the body. For example, albumin, or let's say coagulation factor. As a result, the body becomes more susceptible to sepsis due to liver's response to acute phase protein production. Now, you can have secondary amyloidosis anytime you have a chronic inflammatory process. For example, let's say SLE is going to give you secondary amyloidosis. Now, moving on to our next one, which is senile cardiac type of amyloidosis. And we have deposition of the protein called transthyretin. Transthyretin is derived from the protein AF. And you can remember F from old folks, okay? F for folks, old folks, which is giving us a deposition of transthyretin in our cardiac tissue. So let's look at some pictures. Now this is a section of the heart, and you can see how amyloidosis has caused it to become a little bit wider. This white little dots here and everywhere, these are the deposition. This whiteness of the cardiac tissue is due to amyloidosis. This is a gross specimen. This is a histologic specimen of a cardiac tissue, and this really white region in the middle of the pink nice cardiac tissue would be our transthyretin, which is the deposition on the cardiac tissue of the amyloid in old people. The next one I want to talk about is diabetes mellitus. Diabetes mellitus is depositing a protein called amylin, and it's coming from AE. You can remember E because E is endocrine, right? And this is an endocrine problem because we have no insulin from the pancreas. As a result, it's depositing amylin everywhere in the body. Now, the amyloidosis we see in diabetics are around blood vessels. So you can see that these are the capillaries, right? And you can see how you cannot really see the lumen because they're all kind of deposited with the white amyloid all over. So this is commonly seen in diabetes mellitus. So the next one is our medullary carcinoma of the thyroid, and that amyloidosis is due to deposition of ACAL, which is coming from calcitonin. Alzheimer's is deposition of beta amyloid, which is coming from amyloid precursor protein, or APP, and dialysis associated is deposition of beta 2 microglobin, which is derived from MHC class 1 proteins. Now let's complete our studying with some pictures and some histology. This is our medullary carcinoma of the thyroid. This is, you can see the brownish pigment. Those are the calcitonin, right? And calcitonin is depositing our ACAL, which is our defective protein, which is depositing all over the skin right here. I think this is called lichen amyloidosis, which is often seen with medullary carcinoma of the thyroid, and we see amyloidosis all over the skin. Now, calcitonin is released from the parafollicular cells. So if you see histologic section of the thyroid, you should be able to pick out our parafollicular cells. So you can see that this is a nice, beautiful diagrammatic picture. The pink region in the middle is our thyroid follicle, sorry, and then the purple region are our cuboidal epithelium, And the parafollicular cells are kind of in between our cuboidal epithelium. So these are our parafollicular cells. So we're kind of tying everything together here. This is a typical picture of Alzheimer's. So this is obviously a biopsy of a brain tissue after someone died, and you can see the amyloids everywhere. This is the brownish pigment are the amyloids that are often seen in Alzheimer's. This is a very, very common picture. I think we are familiar with pictures like that often. Now this X-ray picture is a picture of amyloidosis due to beta 2 microglobin. This is due to dialysis associated, and you can see this black arrow showing this region is the deposition of our beta 2 microglobin. That's quite obvious in this picture. And the curved arrow you see here, this is breaking of the supraspinatus tendon. We can see supraspinatus being broken here in this picture. It's just an added information in this picture, but really our amyloidosis is seen in this particular region. So let's finish off with liver amyloidosis and also another cardiac. So we can see that all the dark brownish area of amyloidosis is seen all over the liver. This is just a generalized example. I'm not talking about a specific amyloidosis. And here also with congruent staining, we can see that the brownish pigment all over the cardiac tissue is causing amyloidosis, which is quite apparent in this picture.